Hi, class. This is going to be a short run-through of the fifth paper entitled Actin Filaments and the Growth, Movement, and Spread of the Intracellular Bacterial Parasite Listeria monocytogenes by Tilney and Portnoy. Before I get into the methods and then the results, I want to show you this film, which I'll also include in the lecture unit 5 on actin.
This mammalian cell has been infected with pathogenic Listeria monocytogenes. These bacteria move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake. Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path. If we look closely, we can see a bacterium divide inside the host cell. Immediately after separation, the two daughter cells assemble their own actin tails and start moving about. These bacteria can also form actin comet tails and move in cell extracts. Here, the bacteria are expressing the green fluorescent protein, and actin is labeled red with a fluorescent dye. The dynamics of the actin tails that propel the bacteria through the cytosol can be modeled based on known biochemical and physical properties of actin and actin filaments.
Okay, back to the paper. This is from the very last figure in the paper, or it is the very last figure. And it shows, based on a series of static EM images, what they're proposing to be the progression of Listeria infection into a host cell, beginning with phagocytosis, contact of the Listeria with the host cell surface, induction of phagocytosis into this phagosome, and then subsequently dissolution of the endocytic membrane surrounding the bug by release of what's called a hemolysin. And once it's fully released, it's in the cytoplasm. And sometime later, it acquires this mat of surrounding fibrillar material, which in this paper was identified as actin. Sometimes you see cells dividing, and the daughter cells are acquiring this mat of actin. But in that same time point, they also observe these bugs with a polarized tail or comet tail-like structure of this fibrillar material, which presumably was pushing these bugs to make these cellular protrusions, which, if there was an adjacent cell nearby, would actually invade the acceptor cell, and ultimately the acceptor cell, the next cell to be infected, will phagocytize that, and you have an outer membrane. There's a two-membrane structure shown here, which consists of the acceptor cell membrane and the protrusive invading cells membrane or infecting cells, which ultimately, eventually, the inner membrane, once again, due to secretion by the bug of hemolysin, it's a bacterial enzyme, which incidentally is most active at the acidic pH of the endosomal compartment. And finally, release the bug, captivity. In fact, you delete the gene for hemolysin, you get up to here, but you can't get further transmission because it can't get out of the phagosome, and eventually the bug will be killed by fusion with lysosomes. So what we're going to do before we go to the results is go through some of the key methods. Oh, I just wanted to point out that it had been known at this point that this bug was capable of transmitting from cell to cell in a variety of cell lines that have been examined. So that had been done before. What hadn't been done before to figure out what was the cellular basis for this transmission. Also, as I'm sure some of you know, this bug is a pathogen that's transmitted in contaminated milk products. For example, cheese made from unpasteurized milk. And you get bad diarrhea for the most part, unless you, although it can be lethal to pregnant women and neonates. So let's talk a bit about the key methods. Method one, they were using tissue culture cells of a macrophage cell line. That's important because these cells are capable of being macrophage derived of phagocytosis. And in terms of infection, this is the whole experimental strategy here, is that I want you to realize that this is yet another example of a pulse experiment. They're infecting for 30 minutes, then washing out all the non-internalized bugs and culturing the media for various times in media that contains the antibiotic gentamicin, which will kill any bugs that were bound to the surface but not yet internalized. So you don't get a bad chase. In other words, you're not going to get infection after the first 30 minutes. And summarized here, you infect the macrophage cells for 30 minutes, the pulse. Then you wash out the bacteria and replace the medium, fresh medium with gentamicin to prevent any further invasion of bugs. And then at various times, you fix and process for transmission EM, thin section transmission EM, at 0 time, that is right at the end of the pulse, or for 90 minutes, or 200 minutes post-pulse, the chase. Another key experimental used in this study is the drug cytochalasin D. Cytochalasin D is a drug that inhibits actin assembly. So the other key thing is it's membrane permeant, so you can use it to inhibit actin assembly in living cells. And it inhibits assembly by blocking actin monomer addition to the plus or preferred assembly end of a growing filament. Other key experimental strategy was the use of what is called in the subfragment 1 of myosin. That's the head neck region of myosin. It's the region that binds to filaments. And in this context, they're using S1 decoration. That's a jargon term, but what decoration refers to is the bind of a S1 to each monomer of an actin filament in the absence of ATP, give characteristic arrowheads, which I'll show you in a second. So the key for the use of S1 here was Tilney suspected that the fibrillar material that he observed in these EM studies was actin, and S1 has been traditionally used to identify unidentified filaments as actin or not, because myosin is a very specific, it binds to actin filaments and to only actin filaments, nothing else. So just to put S1 detection in context, here's an actin subunit. It's polarized. This is plus end. This is what actin filament look like. They look like 2 strands of pearls with the twisted with the helical repeat, about 14 of pearls or actin units per helical repeat. This is the same thing, but putting the atomic structure of actin in this model. And this is how you use one. You take myosin, you clip it, in the case of S1 with a digestive enzyme, papain, and you get these single-headed myosin heads, which will bind to actin with high affinity. And they do so because they form an arrowhead type configuration. Here's the structure of the myosin neck domain. And this is an actin. These are all negative stain EMs that I'm going to show you. This is a naked actin filament. This is a mine decorated filament with the arrowheads, that is the point end or the minus end, going in this direction. It's a little clearer in these negative stains where you've got arrowheads, which are actually consisting of, it's a two-dimensional projection of a three-dimensional helical array of myosin heads from helix point to helix point. So this one, the pointed end is going in this direction. The plus or barbed end is here, and this is going in the opposite direction. There's 1, 2, 3, 4. So that's what you're looking at in the thin section, not with nearly this resolution, but it's a thickening. If you go back here, it's a thickening of the filament. It pretty much doubles the diameter of the filament once you add the myosin heads. And here's a model of this, which shows you the nature of these arrowheads. It's not a single myosin head making an arrow, it's 14. So you got, this is the point and the barb, where the minus plus end. This is the minus and the plus end is going off the screen there. And this is the plus end pointing in that direction. So now let's go to the figures. The first figure is looking at a variety of what are stages of infection during the first 30 minutes of infection. And from this, they assemble essentially a progression. But it's important to remember, and it is one of the flaws of the paper, that these were all at the same time point. So the presumed first event is the binding of a Listeria to the surface of these macrophages and the formation of the phagocytic cup, which we talked about in Unit 4. The second presumed progression is the full internalization of the bacteria into a phasosome. They also use the term endosome in the paper. Later, you see fusion of this primary phagosome, fusion events with glycosomes, which contain these vesicular material. And then at a later time, they presume, but remember, these are all from 30 minutes. You see dissolution, but release of the bug hemolysin to dissolve that endocytic membrane. This is in longitudinal section. These are bacilli or rod-shaped bacteria. This is one that's about to divide. And here you can see one in cross-section with the membrane partially dissolved already. And finally, even in this 30-minute time point, they see bacteria that completely dissolved and escape from their endocytic membrane. This is a longitudinal section. It's a cross-section of a bug. Now, why are you seeing so many different stages? My guess is that this early event is a bug. Remember, this is continuous infection for 30 minutes. And this is a bug that may have just contacted the cell at, let's say, 25 minutes. This may have been a bug that contacted the cell at, let's say, 15 minutes ago. And maybe this is a bug that's infected even sooner at the beginning of the experiment. And these guys maybe were the ones to get in first, maybe at 1 or 2 minutes. So it's important to note that these aren't points, they're all from 30-minute fixation. And so what they've done is assemble a progression of events. But when you think about phagocytosis and the sequence of events that occurs, it makes a sense that their assumptions are probably correct. So here are the stages they saw after 30 minutes of infection: phagocytosis, complete internalization, dissolution of the endocytic membrane, and release. This is the next set of figures. And these are cells that were infected, once again, for 30 minutes, but then incubated for an additional 90 minutes after fixation, before fixation. And what you see here is that they're now all completely free of the endocytic membrane, and they've acquired, in the previous example, the free bugs had no fibrillar meshwork or mat associated with them. But here, all the bugs at this time point have this fibrillar mat. This is a longitudinal section. This is the same thing in cross section. And note the diameter of this fibrillar components compared to what they're doing here. This is the same time point, but the cells were first permeabilized with non-ionic detergent to allow addition of S1, myosin head and neck fragment. And as you can see, the fibrillar mat now, the filaments that make up that mat appear much thicker, and that's because of the myosin that's been bound along the actin filaments. One thing to note though, except for a few filaments where you can actually determine the polarity, the direction of the arrowheads are pointing, presumably because these, in fact, we now know from subsequent studies that these filaments are just very short and disordered. And so it's very hard to discern their polarity because they're just a jumbled bunch of very short filaments. So this is in longitudinal section, and this is in cross section. So compare the mesh here to that, and you can see the substantial thickening of the filaments. This essentially provided definitive proof that the material seen here is composed of actin, and it has to be host cell actin because bacteria do not express actin. I just wanted to add this one observation, the figure legend that goes with this. Of interest is that most of the filaments, whether longitudinal or transverse section, appear to be cut in transverse section. That means, that means that these filaments are not in a bundle of aligned parallel filaments. They must be all mixed up at all sorts of different angles. So this summarizes the progression up to 120 minutes here. The bugs are all free, and they have this uniform meshwork of what we now have identified as effect around them. And then let's go on to the next time point, which is 30 minutes of pulse infection and 210 post infection for a total of 4 hours. And they see a number of configurations, cells that are dividing and acquiring their own mesh of actin, but they also see cells that are developing a polarized array of actin pointing in one direction to make a comet tail. And down here is an example. Remember, these are all from the same time point. Is a cell that has already generated one of these very long actin comet tails. This must be very dense and presumably cross-linked, because you notice organelles adjacent to the comet tail are extruded from this volume here. We now know that's because all these filaments are cross-linked with an actin filament cross-linker to make this a very dense mesh that's going to prevent the movement of, for example, ribosomes or polyribosomes into that space. These kinds of structures were also observed after 4 hours. These protrusions of a bug at the tip of the protrusion of the cell membrane, the host cell membrane, followed by this comet tail. This is read from here to here at higher mag. There's the tail end of the bug. And I want to point out is that this fibrillar material, these actin filaments, do not look like a bundle. They look sort of tiny little bits of stuff. And one of the concerns when this study was presented is that we knew from studies done in the Tilney lab actin filaments are very sensitive to disruption by osmium during the fixation protocol. And so that was one concern, that maybe this was a bundle of actin filaments that had been artifactually chopped up by over-osmication. But as sort of a really side-by-side control, this is a microspike or a microvillus, and it's hard to see at this mag, but the filaments are parallel and nicely in the structure right next door to one that has these short filaments. This is the same protrusion type structure, but this is from a cell that's had its membrane removed so that you could add the S1, and you can see the filaments are much, much thicker in diameter, a little bit more distinct. But the other key thing is that once again, they're at all angles. There's no parallel array of filaments emanating from here. These are just short, what we now know are very short, cross-linked and plus end capped filaments that comprise this posterior part of the tail. This is also at 4 hours. Here's one of these cells forming a protrusion, abutting and making contact with an adjacent cell about to be infected. Here's a cell, presumably a similar protrusion, but got all the way in and is now phagocytosed. It's hard to see at this mag, but there are two membranes. There's the outer infected cells membrane and the inner invading cells membrane. And this is the same thing in cross section here. And you can see the two membranes here, but already the inner invading cell membrane, protrusion membrane, has started to dissolve because of release of hemolysin by this bug. So in the text of the paper describing these figures, I just wanted to read this. This is typical Tilney phraseology. Thus, this insidious beast has managed to multiply and spread cell to cell without leaving the cytoplasm of its host. And here are the stages seen at 4 hours. Stationary dividing cells with a uniform mat of actin. Cells that have developed their actin comet tail, using that common tail presumably to push out of the cell and interact with adjacent cells to effect invasion. So you now have a completely phagocytized invadosome with the bug inside, some residual actin left over, and then at some time later, the bug secretes hemolysin to first dissolve the inner membrane, the invading cell membrane, and then next the invaded cells endocytic membrane to release the bug, which is then free to go through another whole round of this and infect the next cell down the line. The big question, though, is this actin assembly mediated by the bug required for this cell-to-cell transmission? And the way they got at that question is shown here. And to determine whether actin was required cell-to-cell invasion or infection, they incubated cells after infection cytocalase to inhibit actin assembly. And the result was quite clear. There was no cell-to-cell transmission, indicating that yes, indeed, it's the pushing of bug out of the primary cell, infected cell, into tanks of the adjacent cell that drives and is necessary for infection. And then the results are shown here. These are two just bright field. These are histologically stained cells that have been infected in the absence or presence of cytocalase D for 7 hours. In the control, which is on the left, you see bugs in all the adjacent cells, these little rods of the bacteria. And the other key thing is in this cell, it's quite clear. This is a nice flat cell. The bugs are out in the cell periphery. But in cytokalasin, this is an infected cell prior to the addition of cytocalase D. And these are adjacent cells. There's no transmission of bacteria to adjacent cells. And the other thing is all the bacteria are perinuclear, sort of huddled towards the cell center, presumably because they can't move around and jet ski through the cytoplasm with their actin column because it's not forming because of the drug, cytochalasin D. This is a thin section EM of a cell that was, I think, 3 hours in the presence of cytochalasin. And as you can see, they have used their hemolysin to get out of those double membrane or single membrane endosome membrane, endocytic, the membrane that's surrounding them, but totally free even after 3 hours of that actin meshwork. And that makes sense because cytokine is going to prevent the bug from driving and recruiting host cell actin to its cell surface. So this, once again, is the whole progression. And the reason this is an insidious beast is that by having this ability to go from cell to cell, it doesn't have to get released for infection, where it can be attacked by the cell's immune system, as well as by macrophages and other scavenging cells that respond to infection. This is really a very remarkable method to avoid host cell system. Now, since this initial study in 1989, this set of discoveries that actin was driving this kind of protrusive activity prompted a huge amount of interest in this as a model system. And so as a result of work from a number of laboratories, we now know a lot more about how this is working. And this is summarized in this diagram from a fairly recent review. Listeria expresses on its surface a bacterial protein, which has been named Act-A. This protein, in turn, recruits profilin actin, we'll talk about in Unit 5, as well as this activator of ARP2/3, which is an actin nucleator. And that's essentially the motor to drive the common tail. You get assembly of these actin filaments. They rapidly become caps. We'll learn the same thing happens during lamellopodial extension by actin assembly. And they're also cross-linked by this protein called alpha-actinin. And then the tail eventually gets chopped up, as we'll learn in Unit 5, by this severing protein called cofilin back here. That's it for paper 5.